Hello, you are welcome back to Unit 4, Session 6 of this course. And this session, we will study the third type of muscle, that is a smooth muscle. And we will end the session with a comparison of some features of the three types of muscles. At the end of this session, you should be able to describe smooth muscle cells and also state similarities and differences between smooth and striated muscles. And finally, list locations in the body where smooth muscles occur. Smooth muscle is important and forms part of the lining of the gastrointestinal tract that churn and prepare food through the digestive tract. And also the linings of blood vessels is made up of smooth muscle, and this helps to control the diameter of the blood vessel and ensure the flow of blood through them. The valves that control the passage of gases and fluids in the body are also lined up with smooth muscles. There are other locations such as the walls of the urinary bladder, the uterus, and the erector pili are made up of smooth muscles. Now let's have a look at the nature of the cells of smooth muscle cells. The smooth muscle cells are elongated and are spindle-shaped, that is tapered at both ends, and their size in diameter is much more smaller as compared to the skeletal muscle fibers. So as shown in figure 31, we have a smooth muscle cell or smooth muscle fiber which is elongated and tapered at both ends, indicating the nucleus as well. Also, the muscle fibers making up the single unit muscle are gathered into dense sheets or bands as shown in figure 31. These fibers run roughly parallel and they are densely packed together, usually arranged so that the narrower portion of one fiber lies against the wider portion of its neighbor. These fibers have connections in that the plasma membranes of two neighboring fibers form gap junctions that act as low resistant pathway for the spread of electrical signals throughout the tissue. Smooth muscles are sometimes referred to as plain muscles in that they do not have cross striations, which is a characteristic of striated muscles. And even though they do not have cross striations, they contain similar contractile proteins as striated muscles. And these contractile proteins are the actin and myofilaments, which are arranged obliquely in the cytoplasm, forming a lattice-like arrangement. These thin filaments are attached to punctate structures called dense bodies, and these dense bodies are located at multiple locations within the cytoplasm and attached to the plasma membrane. The dense bodies therefore function as the Z-lines of striated muscles. An electron micrograph of the smooth muscle reveals that the actin filaments are arranged in an orderly manner and attached to the dense bodies that contains protein actinin. And this protein is the same as the Z-band protein as found in the skeletal muscle. The smooth muscle contains much less protein as compared to the skeletal muscle, and also, for instance, the myosin content of the smooth muscle is 20 milligram per gram as compared to in the skeletal muscle, which is 80 milligram per gram. Also, the smooth muscle does not contain troponin, and however it contains other thin filament proteins. Smooth muscles undergo slow sustained tonic contraction, and the contraction is caused by sliding of myosin and actin filaments over each other. Also, the energy that is involved in the contraction is provided by ATP, and in the course of contraction, myosin acts as an ATPase that makes use of the ATP to produce a molecular conformational change of part of the myosin to cause movement. The movement of filaments over each other happens when the globular heads that protrude from the myosin filament attach and interact with actin filaments to form cross bridges, as shown in figure 32. The myosin heads tilt and drag along the actin filament in a short distance, and the heads release actin filament and then changes angle to relocate to another site on the actin filament at a distance away from it. They can also rebind to the actin molecule and drag it along further. The muscle of smooth muscles does not contain the calcium binding protein troponin, and also because of that, contraction is initiated by a calcium regulated phosphorylation of myosin other than the calcium activated troponin system. Also, the smooth muscle is controlled by autonomic nervous system, whereby the skeletal muscle is rather controlled by somatic nervous system. This table gives a comparison of the three muscles that we've discussed so far, being smooth, cardiac, and skeletal muscles, with respect to their distribution, their function, control, appearance, fatigue, and contractions. Study this table carefully in order to understand how these muscles differ from each other based on these particular models or criteria. Under section 6 of unit 4, we studied the distribution of smooth muscles in the body, and we also described the structure of the smooth muscle cells. We also looked at the contraction of smooth muscles and described the mechanism involved, and also finally described some features of the three types of muscles studied so far, as shown in the previous slide.